Good evening, folks. Thank you for coming out late in the year, in the almost winter, December, a rainy, cold evening, and we appreciate it. I'd also be very much remiss if I didn't thank Dr. Raboli, who allowed this to be the first. I think this is the first time not only are we permitted to eat in this room, but actually the food is actually served in this room. So I think that, I'm appreciative of that because our NPR projection system is not working properly, so we had to move it in here. I do ask, though, if you don't mind at the end, if you wouldn't mind bussing your table on the way out. Before I introduce the speakers this evening, I just want to say a couple things about the Faculty Development Research Workshop Series. We have been doing this now for about 4 years, a little over 4 years. Ostensibly it was to help junior faculty learn new research skills, but it quickly morphed into providing new topics for those who are more seasoned faculty who want to learn new skills. That's what tonight's evenings will be focused on. And more recently, we've been trying to extend it to more junior people, including residents, also the healthcare extenders, the APNs, and the PAs in the health system, as well as nurses. We're always looking for topics. If you have any, please let me know. We have a number of slots still available for the 2020 year, and we'd be glad to host those for you as well. So this evening, we're quite fortunate to have Doctors Jean-Pierre Issa and Jaroslav Jelinek from the Coriell Institute for Medical Research, our neighbor here. Again, I think this is the first time we've ever had someone from Coriell give a seminar, so I thank you, gentlemen, for agreeing to do that. And also I want to thank the big turnout from the folks at Coriell. Some of you I recognize from the Cori meetings and from your biweekly seminar series, as well as the very large turnout from the Department of Pathology. So thank you, and as well as from the Cancer Center. But pathology really turned out this evening, so I appreciate that. So Dr. Jean-Pierre Issa is probably almost everybody here knows, is the new president and CEO of Coriell. He's a world-known expert in the study of epigenetics, and we're going to hear a lot about that this evening. He came to us from Temple University's Fels Institute for Cancer Research and Molecular Biology. Prior to that, he was Chief of Translational Research at the Department of Leukemia at the University of Texas MD Anderson in Houston, Texas. He has many publications, grants, and awards, and I'd just like to mention some of the awards that he's received. The Sidney Kimmel Foundation Scholars Award, the Faculty Achievement Award in Basic Research from the MD Anderson, the Rosenthal Award from the American Association for Cancer Research, and he has been named an American Cancer Society professor as well. He received his medical degree from the American University in Beirut, Lebanon. He did his internship and residency at Good Samaritan Hospital and Johns Hopkins in Baltimore. So before I turn it over to him, let me just introduce Dr. Jelinek, if you don't mind, get that over with, the formalities. Last but not least, though, Dr. Jelinek came along with a number of Dr. Issa's colleagues from the Fels Institute. He is Coriell's Chief Research Officer. He also was a visiting research professor at the MD Anderson Department of Leukemia in Houston. He has over 120 peer-reviewed articles. He received his medical degree, excuse me, and his doctorate in physiology from the Charles University in Prague in the Czech Republic. So this evening, we're going to back and forth, so we'll start with Dr. Issa giving the introduction to the area of epigenetics for the clinical investigator. Thank you.
Right. Thanks very much, Harry, for this kind introduction and also the invitation to speak here. As you mentioned, we're almost literally next door, less than a block away. We'd love to have collaborations with people at Rowan, at Cooper, and we have some already ongoing. We'd love to see more, and so this is essentially. We're happy that people from Coriell are here to speak. Actually, it's an exchange day because our seminar speaker at Coriell today was from this medical school. So today is an interesting day in this respect. So we're going to tell you a lot about epigenetics today. And the way we're going to do it is I'm going to give you a brief background on why it's important and how we think about epigenetic from a sort of a bird's eye view. Then I'll turn it over to Jaroslav, who's going to tell you a very more technically oriented talk about how one measures these kinds of things, both in the lab and potentially in the clinic. And he'll give you some examples of the work that we have done together over the past few years. And then, time permitting, I will come back and tell you about some of the more clinical applications of epigenetics, focusing in particular on drugs and clinical trials with drugs that affect epigenetics. So the end will be more directly clinically oriented here. So just as a primer on epigenetics, it's important that we define it carefully and that you all understand what we mean when we talk about epigenetics. The term epigenetics was coined by a developmental biologist, Conrad Waddington. Epi meaning above the genome, so this is where the name comes from essentially here. And this was work in the 1950s, and if you recall, the beginning of the 20th century, no one really knew what the genetic material was going to be. It was thought actually to be protein rather than nucleic acids. When it was discovered to be nucleic acids, there became a mystery at the time, and this is what Waddington was addressing, which is that it was clear that nucleic acids did not have a lot of complexity, certainly not the complexity of proteins. And then it was poised that every single cell in the body would have the same code, would have the same genetic code. This was even before it was deciphered, of course. But then Waddington was dealing with the phenomenon of, well, how come you have 200 different cells and tissues in the body, and they all have exactly the same genetic code? There must be an additional code that works on top of the genetic code that is responsible for this phenomenon of cellular identity. And so at the time, he had no idea what it would be, but he hypothesized that there must be a code other than the genetic code to determine cellular identity. And he thought about cellular identity as these hills and valleys in this case. And the idea was that you would start, let's see, is this the pointer? Yeah. So a cell would start here as a completely primordial, undifferentiated cell, and that it would travel down, and it might travel up a hill, or it would travel in a valley here, and it would end here in a terminally differentiated state with no going back. And he proposed that these hills and valleys essentially was the epigenetic code, which at the time we didn't know what it was, but now we essentially know what it was. Now, this all seems fairly theoretical to you, so I'd like to give a very concrete example. So these are my kids many years ago. And basically, just to tell you that genetics, of course, determines essential things, like they determine the fact that they are female, that, they're essential characteristics here. But if you look at a given human being, the eye, the skin, the teeth, the hair, the inner organs, all have the same genetics, yet these tissues are very different and they're very stably different. So liver doesn't turn into lung and so on. And so this code is a very stable code. And the biggest misconception in the field is people who think that epigenetics is very plastic. In fact, it is not plastic. Once it's established, it is supposed to be very stable. This is what determines cellular identity in humans here. Now, this was the beginning of epigenetics, but as it evolved, scientists discovered different phenomena that essentially had the same molecular underpinning. And for science and medical purposes, one of the most interesting one is this phenomenon of phenotypic variation that has an epigenetic underpinning. So what you see here is a mouse model. This is the pseudo-agouti mouse model here, which was worked out by Emma Whitelaw's lab in Australia. And you see 6 mice that are lined up that are relatively, there's nothing unusual about the mice at the here. The biggest unusual thing is that this yellow mouse and this brown mouse are exact identical twins. They have exactly the same genotype. You cannot find a genetic difference between the two, yet the phenotype is very different and again very stable. When this mouse grows older, it will become obese and it will die of cancer. When this mouse grows older, it will remain lean and it will likely die of an autoimmune disease. And so you see there's exact same genetics, yet there's a very different phenotype, and including disease phenotype in that case. And that epigenetic difference turned out, now we know what it is. It's a DNA methylation difference between these particular mice here. So we are now interested not just in development and cell identity, but potential phenotypic variation that is encoded in the epigenome here. And for people who are interested in other examples of this, this is one of the beautiful examples of that in nature. Cat lovers will recognize this cat as a relatively rare type of cat called the calico cat. And the calico refers to the color of the cat, which is this mixture of black here and blonde here, and the white being the background in that case. And what's unusual is that this mixture of color in calico cats almost guarantees you that this is a female cat. In fact, cats that look like this are about 3,000-fold more likely to be female than males. And the reason they are like this is because cats can, these types of genetics, the genetics of these cats are they can be either all black or all of this color, except in females, the gene that determines this color is on the X chromosome, and half of the X chromosome, half the cells have one X chromosome, the other half of the cells have the other X chromosome expressed. So this is the phenomenon of X inactivation, which results in this epigenetic skin color state, or hair color state in this case here. So this variation here is also an epigenetic variation. There are other examples of that in nature that are quite interesting in that case here. Okay, so cellular identity, phenotypic variation. We now understand that there's also probably a third very important property of epigenetic inheritance, and that is the same enzymes that are responsible for this and this are also responsible for keeping the noise down in cells. As you know, human cells are composed of mostly repetitive elements. The vast majority of our genome does not code for protein, but essentially is composed of viruses and other repetitive elements and intergenic areas here. And these elements, which include DNA transposons, LTRs, retroviruses, and so on, for the most part have to be kept shut down because their expression could be deleterious, it could kill the cells, it could introduce mutations, and the way these are kept off is using the same epigenetic mechanisms responsible for cellular identity and for the phenotypic variation that I described earlier here. And this becomes relevant for people who are interested in disease biology because these may actually contribute to autoimmune diseases, and this is also relevant to the use of drugs that change epigenetics, because by affecting epigenetic enzymes, not only can we affect the phenotypes I described to you, but we can also affect expression of these repetitive elements here. Okay, so this whole phenomenon of epigenetics was completely mysterious in the 1970s and early 80s. I remember when I was training as a postdoc, there was a whole issue of Cell dedicated to this, and it was called the mystique of epigenetics, because for 30 years people knew about these phenomena, but there was really no real explanation for how it happens. And then in the 1980s, this entire epigenetic code was deciphered, 80s, 90s, and Jaroslav is going to talk a lot about that, but ultimately everything relates to how the DNA package is packaged inside the cell. So if the DNA is relatively open, then the gene is expressed. If the DNA is very, very tightly worn down, then it is not expressed. And so packaging DNA ultimately is how the cell achieves whether a gene is on or off. And this is done through these enzymes that you're going to hear about later on here. Now, since this is a clinical talk, or not clinical talk, but there are some clinicians in the audience, I wanted to mention that in medicine, we are very interested in epigenetics because it is associated with a number of normal and abnormal phenotypes. So I mentioned development and differentiation. Of course, stemness, or stem cell versus non-stem cell, is an epigenetic phenomenon. X inactivation in women. Imprinting, which is a phenomenon where a gene is expressed differently depending on whether you got your father's copy or your mother's copy. This is a really interesting phenomenon, imprinting, which is an epigenetic phenomenon and is probably evolved with a single function, is to make sure that babies come from a male and a female rather than two females or two males. Theoretically, you can make babies just with two females. The genetic diversity will not be the same. And imprinting is what ensures essentially sexual selection in that case. We talked about natural variation. Now, given how important epigenetics is to development, you won't be surprised to know that we now know a number of childhood developmental diseases that are directly linked to abnormal epigenetic function. In fact, the most common form of mental retardation in boys is the Fragile X syndrome, which is due to abnormal epigenetic regulation of a gene called FMR. The most common form of mental retardation in girls, in young girls, is the Rett syndrome, which is due to a mutation in an epigenetic regulator called MECP2. So these epigenetics play a very important role in childhood developmental diseases. You'll hear a lot more about aging and cancer in Jaroslav's talk, and I'll come back to talk about it a little bit later as well. I will mention to you the potential of reprogramming and anti-cancer drugs as well after you hear some more about the basic mechanisms of epigenetics. And this is when I'm going to turn it over to Jaroslav to talk about mechanisms and his work here. Is that yours?
Yeah, that's mine.
You want to turn on your mic, and you know how this works? All right.
All right. So DNA methylation is adding two methyl groups here, and I added them to Coriell logo because we so DNA methylation is an evolutionary old mechanism. Not long ago, after primordial soup sprang to life here in Camden, bacteria developed used DNA methylation as a protection device against invasion of viruses. So the way it works, that bacteria make DNA methylase and restriction endonuclease, and bacteria methylate their own DNA, and it protects the DNA against restriction endonuclease. And the invading DNA doesn't have this protection, so it's cleaved out. Multicellular organisms repurpose these mechanisms for regulation of gene expression. So here is DNA methylation as an epigenetic mechanism, and here is modification of histone tails, and these modifications dictate whether chromatin is closed and inaccessible for gene expression or open and accessible. So one genome, many epigenomes, 200 or hundreds of different cell types that are governed by different sets of genes that are expressed, and this is dictated again by DNA methylation and histone modifications, which could be acetylation, methylation, DNA methylation in mammals or in vertebrates happens in the CpG context. CpG means cytosine followed by guanosine linked by P by phosphodiester bond. And those CpG sites are relatively rare. And we have about 28 million CpG sites in our genomes, and most of them are scattered and methylated. And there is an exception of CpG islands that are dense clusters of CpG sites that are typically unmethylated, and they are present typically at the transcription start sites of genes. About half of our genes have those CpG islands at their transcription start sites, and they are not methylated. So we have only 28 million CpG sites in 3,200 million bases. Is that right? So when we do simple calculations and calculate the probability, how many CpG sites we should have based on 16 possible dinucleotide combinations, we should have about 200 million CpG sites. We have only 28 million, which means there is a 7-fold depletion. So why is that so? The main reason could be that methylcytosine is mildly mutagenic. Simple deamination of this base converts methylcytosine to thymine, and C to T transition is the most frequent mutations found in cancers. So this Nature paper defined 20 different signatures of mutations, and this C to T transitions shown here is the most frequent mutation. It happens in the germline and it happens in somatic cells. P53 mutations are frequently C to T transitions. CpG methylation is an epigenetic mechanism that is relatively stable. So 80% of CpG sites have the same methylation pattern in all cell types. About 20% of CpG sites are plastic, and they are different in different cell types. This graph shows principal component analysis of DNA methylation patterns in different cell types, and these are primary cells, and they are very close to each other. There are some blood cell types that are very close. In vitro derived cells are somewhat different. Sperm methylation patterns is very different, and this is a tumor cell that has really different methylation patterns, and these would be tumor cell lines that are very distant. So again, this is the typical pattern: unmethylated CpG islands and methylated CpG sites that are depleted and scattered. DNA methylation is context dependent. So promoter CpG islands are typically not methylated, which shows this green color. Transposons, repetitive elements are typically methylated. Gene bodies are typically methylated, and those plastic sites could be non-CpG island promoters, non-promoter CpG islands, and in enhancers. How do we detect DNA methylation? So bisulfite conversion is a gold standard method for detecting and measuring DNA methylation. It works this way. So cytosines, if they are not methylated, are converted by bisulfite to uracils. And after PCR amplification, this uracil, which is not a DNA base, is changed to T-thymines. Methylcytosines are protected from bisulfite conversion, so after bisulfite they stay methylcytosine, and after PCR amplification they become cytosine. So this is an example of a methylated CpG sites that remains after PCR amplification as CpG sites. There should not be the methyl group here because PCR amplification doesn't reproduce methylation. Cytosines that are not a part of CpG sites, they are typically unmethylated, so they are converted to uracils and they are converted to Ts. So disulfide conversion creates SNPs or single nucleotide polymorphisms at the sites of CpG islands, and we resolve those polymorphisms by sequencing. Restriction enzymes that are sensitive to DNA methylation can also be used for detection and measuring of DNA methylation, and we have used those. This is an example of a restriction-based method for detecting DNA methylation and next generation followed by next generation sequencing. And it illustrates this 80% of stable and 20% of variable methylation. DNA methylation in CpG islands in the blood versus liver is either unmethylated or completely methylated, and very few CpG sites far in between. CpG sites that are not part of CpG islands, they have more variability, but still only 16% of those dots have different methylation in the blood, in the normal blood and normal liver. This is shown in rat tissues. So this is frontal cortex, this is spleen. So 3 different specimens of frontal cortex have practically identical DNA methylation in CpG islands and in CpG sites that are not present in CpG islands. And there is very little difference in CpG islands, and most of those differences between brain and spleen tissues are in CpG sites that are not part of CpG islands. In healthy tissues, we have CpG islands that are not methylated and other CpG sites that are sparse, they are methylated. In cancer, this pattern is disrupted and switched, so there is global hypomethylation, so those CpG sites that were methylated in repetitive sequences in gene bodies may become hypomethylated, and CpG islands become hypermethylated, and this has functional consequences because this hypermethylation of transcription start site, CpG island at transcription start site prevents gene expression, and something in between is normal aging or diseases like inflammation, where there is this erosion of distinct unmethylated and methylated states. So some of those CpG islands, sites at CpG islands may become methylated, and there is some hypomethylation elsewhere. So we can see it in the mouse model in these young mice. So these are 5 different young mice. Blood cells, DNA methylation is practically identical in those inbred mice. In middle age, you can see that this cloud is somewhat wider, so it means that there are more inter-individual differences, and it's accentuated in the aged old mice. So these would be mice that are more than 2 years old, and you can see spreading of those methylation differences. And it's recapitulated in humans, so this is human blood, this is human cord blood, practically identical methylation in different cord blood samples, somewhat spreading in the middle age and more spreading in the old age. Shinji Megawa and colleagues in our lab examined lots of regions for age-related methylations and found DNA methylation drift in the mouse, in rhesus monkeys, and in humans. These are different genes, CpG islands, and you can see a gradual increase of methylation at those CpG islands. And what was interesting that this rate of methylation was related to the lifespan of the species. So the mice that have a lifespan of about 3 years had this rate of methylation higher, and monkeys in the middle, and humans the slowest rate of methylation change. And more, this methylation drift could be reversed by caloric restrictions. It was shown in monkeys and mice. This is hierarchical clustering of DNA methylation patterns in promoter CpG islands, promoter hypomethylation, and non-promoter CpG islands. The main message is here. So this white rectangles are young mice. These are caloric restriction old mice, and these are old mice. So caloric restriction mice, based on their methylation pattern, cluster with the young ones, and this is recapitulated in monkeys as well. We are looking for markers of age-related methylation drifts, and there are two things. There is chronological aging and biological aging, which may or may not be identical. So this gene has been widely reported in the literature as a good indicator of chronological aging. We looked in using next-generation sequencing and looking at hundreds of thousands of CpG sites, finding CpG sites that would be drifting with aging. This is an example of some CpG sites that we found. DNA methylation drift is affected by aging, but it could be also affected by inflammation. So this is a mouse model where genetic knockout of interleukin-10 leads to inflammation, and it's associated with increase of DNA methylation. And the largest drift was still found in aging mice. We also looked at epigenetic changes that would be associated, that would be found in a chronic pain model, and this chronic pain model was in collaboration with Dr. Pan's lab at MD Anderson. They did spinal nerve ligation in the rat model, so these spinal nerves were ligated, and we looked at changes of gene expression, DNA methylation, and histone methylation in dorsal root ganglia. And we also looked at, or the collaborators looked at the chronic pain by hyperalgesia caused by pressure or tactile allodynia. So this is how it was detected. And this spinal nerve ligation resulted in development of chronic pain about 3 weeks after the procedure. And when we looked in dorsal root ganglia, we found gene expression changes in potassium channels here and in other differentially expressed genes. So there were some genes that were downregulated or upregulated after 3 weeks after spinal nerve ligation. But what was interesting that an inhibitor of histone methyltransferase. This UNC drug inhibits G9-8 histone methyltransferase, and it changed or it normalized the gene expression changes and also relieved the chronic pain. We also found DNA methylation changes after this surgical procedure and 3 weeks after surgical procedure and chronic pain. And there was general hypomethylation, which is shown here by 2 methods, restriction-based and bisulfite-based method. So this was chronic pain, this was aging, but cancer is the disease that is associated with greatest DNA methylation changes or epigenetic changes. This is an example of patient with acute myeloid leukemia, and DNA methylation is compared between normal white blood cells and white blood cells from myeloid leukemia, and this shows great hypermethylation at many CpG sites in CpG islands, and also hypomethylation of CpG sites outside of CpG islands. DNA methylation changes or DNA methylation aberrations are associated with prognosis. In the relapse of acute myeloid leukemia, we found increased DNA methylation at multiple genes. DNA methylation and those genes was also associated with shortened survival in patients with myelodysplastic syndrome. It was associated with shorter survival in chronic myelogenous leukemia, but surprisingly, we found that in acute myeloid leukemia, several genes that were hypermethylated were associated with much better survival. So we looked in a larger cohort of acute myeloid leukemia patients, checked DNA methylation at thousands of CpG sites, and found distinct groups based on DNA methylation patterns. This is hierarchical clustering showing patients with decreased methylation showing in green, greatly increased methylation shown in red. This is for comparison in normal controls, and somewhat methylation changes that we could see in many other cancers. And this was demonstrated with different survival rate. So these patients with greatly increased methylation, CpG island methylator phenotype, originally postulated by Dr. Issa, had best survival, and those hypomethylated patients, survival was in between, and this median group with moderately increased methylation had the shortest survival. We looked also at methylation signatures and found increased mutations of known genes IDH1, IDH, isocitrate dehydrogenase 1 and 2, which explains hypermethylation, but there were patients that didn't have mutations in these IDH genes, and they had the best survival. So in summary, DNA methylation is crucial for development, differentiation, tissue-specific gene expression, and chromosomal stability. It occurs in cytosine followed by guanosine, so at CpG sites. Methylation of promoters at CpG islands prevents transcription. methylation enhancer is dynamic, and it's the most flexible methylation, and methylation patterns are disrupted in aging and most in cancer. Lots of people participated on this work in our lab, and we also collaborated on this chronic pain model with a PAN lab at MD Anderson. And this is a picture when we were transferring from Temple to Camden. It was last February. It was very cold. You can see that. So thank you.
Thank you, Jaroslav. So we'll keep questions to the end. I'm going to build on what you have heard and tell you a little bit about some of the translational work that we have been doing here. And I want to go back to one of my favorite examples of epigenetics here. And it is this example. So this is the queen bee versus the worker bees here. You can see in this colony, mostly worker bees, and then the queen bee that is fluorescently labeled in the middle. They're all genetically identical. They have exactly the same genomes. The queen bee, of course, is the queen. But beyond that, the queen bee lives for many years. There's been examples of queen bees living up to 30 years. The worker bees live for months at most. And so same genome, very, very large difference in survival and in lifespan in that particular case here. So this fits with the aging phenotype that Jaroslav was talking about, potentially that aging has an important epigenetic component, and many age-related diseases do here. But the other thing that's fascinating is to ask the question, well, how does this come about? If it's all the same genome, how do you get a queen bee versus a worker bee? And it turns out that it's all encoded in royal jelly. So queen bee larvae are essentially reared in royal jelly all the time, while the worker bees are only exposed to royal jelly for, I think, a few minutes, and then that's it. They're out of the royal jelly. And essentially, it's the royal jelly, which is an environmental exposure in this case, is what determines whether a bee is going to be a queen bee that lives up to 30 years versus a worker bee that lives only a few months. And this just shows you, first, the power of epigenetics in terms of determining phenotypes, but also the possibility that epigenetics can be modified by exposures, by developmental exposures, and so on here. And so the question then is what does this mean to humans? And so what our model is in terms of aging is that we are born with a certain set of epigenetic properties, but that our cells, when we are young, are largely identical. So if you take two stem cells from a young person, they're going to look pretty much the same. But as we grow old, there is a steady accumulation of these epigenetic variants or epigenetic defects. And this is a linear process across the population, but it actually varies quite a bit from one person to the other. And we and others have traced these variants to various things that happen to us. Chronic inflammation seems to increase the rate of change, and that is, of course, associated with mortality. Chronic inflammation is one of the great accelerators of diseases and disease-related death here. And calorie restriction, at least in mice and monkeys, is associated with slowing down of the process. And of course, in mice and monkeys, calorie restriction is associated with greater lifespan. And so our hypothesis then, that is that lifespan within a given species is in part also related to factors that determine the rate of accumulation of these epigenetic defects. And we're interested in understanding these both to measure the rate of disease, but also potentially to measure the rate of age-related diseases. And we believe that you hit a threshold where then the likelihood of a disease becomes higher. It's still a stochastic process. It's not like when you hit that threshold, it happens in everyone, but the likelihood of a certain disease increases greatly, we think, when you hit a certain epigenetic threshold. And one of the projects that we are interested at Coriell is to essentially develop biomarkers of this. And that's one of the collaborative projects that we have at least talked about doing with folks at Cooper, which is develop biobanks of relatively healthy individuals and try to ask, can we have biomarkers of biologic age as opposed to chronologic age? And do these biomarkers of biologic age correlate with diseases? Do they correlate with frailty? Do they correlate with outcomes after surgery, for example, and so on? And so that's one of the projects that we have currently ongoing, and we're developing the tools to do that here. Okay, now, in addition to that, there are diseases in humans that appear to be very, very prominently epigenetic diseases. I actually am a trained medical oncologist, and my clinical specialty was myelodysplasia and acute myeloid leukemia, not completely by chance. It turns out that myelodysplasia is essentially an epigenetic disease. If you ask what causes the myelodysplastic syndrome, which is a pretty devastating disease in older humans, they can happen because of mutations in genes like TET2 or DNMT3A or ASXL1, or they can happen as a result of methylation. But all these genes actually feed into epigenetic regulation. You know, the biggest part of what is wrong with MDS is epigenetic regulation. And the clinicians in the audience who treat these patients will know that MDS is essentially a disease of bad differentiation. It's a disease of it's a cancer of the hematopoietic stem cell that is associated with bad differentiation, and it progresses eventually to acute leukemia. And this is largely an epigenetic disease in that case here. Now, as Jaroslav mentioned, there's also a lot of epigenetic abnormalities in cancer. One can see that disrupted cellular identity. One can see disrupted tumor suppressor gene silencing. One can see disrupted repetitive elements in here. So cancers in general have very prominent epigenetic defects in that case here. Now, one of the questions that then come around all of this is, what of it? You know, can we do anything about this? Of course, we're interested in biomarkers, and he showed you potentially that one can use these tests to stratify patients into those who do well, those who do poorly. But ultimately, can we intervene on epigenetics here? And the answer is yes. And the best example of this comes from nature again. Now, I started this talk by stressing how stable epigenetic changes are and how they are largely irreversible. You know, liver doesn't turn into lung. So we think that once they are fixed in the genome, they may evolve over decades in humans, but it's not something that's going to change from day to day. However, there is one phenomenon that happens during our lifetime that is associated with massive resetting of all of this epigenetic information, and that's during embryogenesis, right? So when you have a fusion of a sperm and an oocyte, really the first thing that happens is a massive decrease in DNA methylation, down to almost zero, essentially. And that happens in the first, 3 or 4 cell divisions. And that's absolutely essential for life. So this phenomenon of epigenetic reprogramming is what makes embryogenesis possible. If you mess with it, you cannot have embryogenesis here. But as a result, most of the epigenetic patterns that come along in adult cells can actually be completely erased, and you reestablish normal epigenetic patterns during the process of embryogenesis, and it looks a little faster for male cells than for female cells, but this happens again during formation of oocytes and germ cells here. So this phenomenon of epigenetic reprogramming is absolutely essential for life. It's essential for embryogenesis, and it is why essentially Lamarckian inheritance almost does not exist, right? Because you would think from everything that I told you that epigenetics would be a prime example of a Lamarckian phenomenon, whereby the environment affects the next generation. But in fact, it does not for the most part, because most of the epigenetic changes that come from exposures and the environment are actually a reset during embryogenesis. Now, I said most because there's a lot of interest in maybe the rare case where something is not reset, and there are some clinical examples of that, for example, grandchildren of people who were born, who had children during famine times in Sweden had a higher incidence of diabetes, for example. And so there is this idea that maybe having kids during periods of famine will reset the epigenome a little bit so that your grandchildren will actually have some disease associated with that. But, you know, these are disease susceptibilities. Probably they are not really major things. So we're still looking at genetic inheritance rather than epigenetic inheritance. And this is because of this process of epigenetic reprogramming. Okay, so reprogramming is fascinating. It's essential for early embryogenesis. It's required for normal development and differentiation. But the reason I'm talking about it so much is because it can actually be done to adult cells. And this was the first animal that was born as a result of epigenetic reprogramming. And people might recall that this is the Dolly sheep. And Dolly was a cloned animal, and cloning is forced epigenetic reprogramming. And the way this was done was by taking the nucleus of an adult cell and dropping it into an enucleated, fertilized oocyte. And at that point, the process of epigenetic reprogramming took hold, reprogrammed the genome of that adult cell, and that cell now became a pluripotent germ cell that gave rise to a largely intact animal. Dolly had some diseases. It actually had an autoimmune disease, and that's one of the things that happens with incomplete reprogramming during embryogenesis. You actually get a first-generation phenomenon in cloned animals where you often have autoimmune or other imprinting diseases. And usually, if these animals then have other animals have offspring, the offspring will not have these defects. And so these are epigenetic defects from the reprogramming process itself that are then completely erased when reprogramming is done naturally. But the point is, it's actually theoretically possible to reverse and reset the epigenome in an adult cell. And so one of the things that I've been interested in for many years now is, what if we try to apply this to cancer cells? You know, can we actually reprogram the epigenome of a cancer cell, and would that lead to cancer therapies? And so the answer is actually yes, and the answer was very humblingly found in literature from more than 50 years ago. So before the genetic revolution, before people knew that cancer was a genetic disease, there was a lot of discussion as to what is the nature of cancer. And there were veterinarians in the 1960s and 70s, and this was actually something. McKinnell is veterinarian from Penn, I believe. I believe he's still at Penn. I think I heard that he still comes occasionally, but that did these experiments where he transplanted nuclei from a cancer, a frog cancer, into a frog embryo, and he reversed tumorigenesis that way. He got normally born frogs that were derived from cancer cells. And this was actually done in mice by Rudolf Jenisch's group. There were a number of different experiments that showed that you can take a cancer cell, force it through embryogenesis, which then reprograms its epigenome, and you erase the cancer properties of these cells. And again, very interestingly, if you look at this 1969 paper, this was quite a while ago, the first paragraph of this science paper says cancer is an epigenetic disease. So that was actually a fascinating, look into a history of science here, because this is something that when I started in the lab and the Balin lab where I trained was saying cancer is an epigenetic disease, we were laughed out of rooms, because everyone knew that cancer was a genetic disease. But anyway, this has changed, fortunately. So the question is, can we potentially do this to people? And, there's lots and lots of questions about, can you do it? How toxic it is? Will this result in, people growing an extra arm or something like that? So there were a lot of concerns early on, but then it was still tested first in mice and then eventually in humans. And we now know that we can achieve some degree of reprogramming of cancer cells. This can be done in the clinic by inhibitors of DNA methylation, inhibitors of histone deacetylation. We are now testing BET inhibitors. These are bromodomain containing proteins that are involved in chromatin resetting here. And there's a lot of interest now in more targeted epigenetic therapy with drugs targeting epigenetic enzymes such as EZH2, DOT1L, IDH1 and 2, LSD1, and so on here. And so in my lab, we've actually been trying to go one step further and saying, can we actually discover new drugs based on epigenetics here? And so this is one of the experiments we did at Temple a few years ago, where a student of mine developed this reporter assay, which we now have at Coriell, where the cells at the baseline condition are not green, but if they are exposed to drugs that reset chromatin, they turn green. So this is essentially a live cell assay for epigenetic reprogramming, and we've done a lot of things with it to discover pathways and to discover drugs. In this particular experiment, we screened an FDA-approved drug library and discovered many drugs that had epigenetic effects. The top drug was already known. It was a DNA methylation inhibitor, so we rediscovered that. That was good. But then we discovered many other things. For example, cardiac glycosides, which are drugs that were developed to increase contractility of the heart through inhibition of sodium-potassium ATPase, actually have very prominent epigenetic effects in our system, and we propose that these could or maybe should be tested as anti-cancer drugs as well. And one of the drugs that was fascinating is arsenic trioxide. This is an example of the cells before treatment and after treatment. You see all these green cells have been epigenetically reprogrammed, at least at this locus. And arsenic trioxide is a component of a Chinese medicine that's been known for hundreds of years to be effective in leukemia. And it turns out that as a single agent, it is curative for a rare kind of acute leukemia called acute promyelocytic leukemia, which is a differentiation disease. And essentially, this one drug can cure that very devastating leukemia, we think in part through epigenetic reprogramming in that case. And more recently, we've taken this one step further and gone back to this model here, where we said, okay, the royal jelly must contain something that affects epigenetics. And in fact, we published a few years ago, Mark Bedford at MD Anderson published in collaboration with my lab that royal jelly contains a compound that has histone deacetylase properties, and so essentially led to the idea that there must be natural compounds that affect epigenetics. And so the next screen that we did, this was the work of a student, a PhD student at Temple, Hang Zheng, who graduated and is now in medical school at Temple. And he screened a natural compound library in the same system, you know, went through thousands of drugs, and eventually came up with this aminothiazole, which we discovered was an inhibitor of a cyclin-dependent kinase, CDK9. This is a kinome assay for inhibition by this enzyme here. And then this drug actually prolongs survival in mice with cancer. And we're developing, you know, we're taking this drug to a new company potentially to try to take it to the clinic as a new form of epigenetic therapy. So we're very excited about the potential for epigenetics, not just as biomarkers of age or biomarkers of disease, but potentially as sources of new drugs. Jaroslav mentioned the use of these drugs to treat chronic pain, for example, and we are interested in the use of these drugs to treat cancer as well here. And so in the remaining part of the talk, I just want to describe to you some of our clinical efforts. So as I mentioned, I'm an oncologist, and I've been involved in clinical trials of drugs that affect epigenetics. And we've learned a few interesting things through these clinical trials. And I thought it might be fun to show you some of these data here. Now, these were the first drugs that were epigenetic drugs and that we worked with. These are ASA nucleoside compounds. So they're very simple. Drugs. This is cytosine here. This is 5-methylcytosine, and these are analogs. This is the ribonucleotide, and this is the deoxyribonucleotide, with the only difference being this substitution here, C2N substitution at the 5 position, which then does not allow methylation at that 5 position and actually traps DNA methyltransferases and results in inhibition of DNA methylation and reprogramming. And these drugs were fascinating because this phenomenon was discovered by Peter Jones in 1979. And this was one of the real classical experiments that proved that DNA methylation is a mechanism of epigenetic regulation, because he treated cells with these drugs, he treated cultured cells with these drugs, and they went from fibroblasts to beating heart or to beating muscle. And so he was able to show really that by changing DNA methylation, you can change cellular identity. Then they entered clinical trials in the 1970s as anti-cancer drugs, and the oncologists concluded that they were too toxic, and they were put on the shelf, and they were actually pretty much ignored through the 1980s and until really Steve Balin's lab, and then also Peter Jones's lab in the early 90s started talking about cancer as an epigenetic disease. And of course, I was in one of these labs, and looking at the literature, I started wondering, well, if it's an epigenetic disease, why won't these drugs actually work? Why do drugs that change epigenetics not work if we're correct? Then there must be something going on here. And so I and some colleagues who, I was working with decitabine, other colleagues were working with azacitidine, and we reintroduced them into clinical trials. And one of the things that I'm not showing you is that it turns out that there's an exquisite dose dependence to these effects, because these drugs, when they were developed in the 1960s in Czechoslovakia, actually in the Czech Republic, they were developed as cytotoxic anti-cancer drugs that block DNA synthesis and induce cell death. When they were taken into clinical trials in the 1970s, they were taken along usual clinical development paradigms, which say the more the better. So, you know, they were given at the progressively higher doses until a very high dose would reach that would not kill the patients, but was tested as an anti-cancer agent. And when Peter Jones tested these drugs, he actually found that as the higher the dose you go, the lower the DNA methylation enzymatic inhibition, because there is a self-fulfilling mechanism that shuts down its activity at high doses. These drugs require replication to work. If the cells sense DNA damage, they stop replicating, and if they don't replicate, they do not reset their epigenome. And so when I started doing clinical trials, I said, let's test this, and we're going to take this drug where there was a known dose that had been given to people, and we said, let us redo these experiments at much, much lower doses. And so I did this trial where we went back to acute leukemias and myelodysplastic syndrome, and we tested the drug at a dose of 50 milligram per meter square, which was actually about 30 to 40-fold lower than what had been given to people before, because at MD Anderson and elsewhere, people were giving about 1,500 milligram per meter square to 2 grams per meter square. And we found this interesting phenomenon is that we saw responses at these low doses, and the responses were actually substantially lower at high doses. And this changed the paradigm a little bit about how we develop drugs in oncology, particularly if we're developing epigenetic drugs, because we argued that to reprogram the epigenome, you need to allow cells to divide, you need to avoid cytotoxicity, and this is what was happening at these low doses here. And you know, to summarize a lot of work, it turned out that these drugs actually work better than chemotherapy. They don't cure the disease, but you see better survival with this hypomethylating drug compared to a matched group treated with intensive chemotherapy. In parallel, the other drug, azacitidine, was taken to randomized study, and this was the survival on azacitidine compared to control. And importantly, both azacitidine and decitabine were being used at doses that were significantly lower than the maximally tolerated doses, and they eventually became FDA approved for the treatment of MDS and now acute myeloid leukemia based on these clinical trials that actually were mechanism-based, where we said we don't want the highest possible dose, we want the dose that changes the DNA methylation the best in that case here. And we saw some really weird phenomenon that remains incompletely understood, which is that in many patients, we treat with the drug. So this is a patient who had acute leukemia. This is the number of blasts in the blood, so they should be 0. This patient had somewhere between 50 and 80% blasts in the blood. we give the patient the first cycle, the second cycle, the third cycle, the fourth cycle, and really nothing happens. This is 5-day cycles, and there's actually a month between each of these things. So 1 month, nothing happens. 2 months, nothing happens. 3, 4, 5. And then after 5 months, you know, as these cartoons say, and then a miracle happens, and we still don't know what happens, you know, the blasts start going away. And we saw this phenomenon in a few patients, and we think that it actually takes time to reprogram the epigenome. And then once we reprogram the epigenome, it then takes time for something else to happen. And the biggest current hypothesis as to what is happening here is that we are overcoming immune surveillance. We're activating the immune system to recognize the cancer cells, that they were tolerant here, perhaps because of downregulation of MHCs, or perhaps because of other mechanisms associated with leukemia, and that the reprogramming made the cells visible to the immune system, and suddenly you see this immune response. There are other hypotheses. The other hypothesis is that we're exhausting the stem cells, is that the stem cells can divide a few times, but then they can no longer divide, and that's why we're seeing this effect. But this is almost never seen. This is not seen with chemotherapy. With chemotherapy, it either works or it doesn't, but we don't see something where it doesn't work for like 4 to 6 months, and then suddenly it works. And that's why we think this is actually different kind of therapy, and this is truly epigenetic therapy. Here, this was one of our patients at MD Anderson in Texas, and this was when the drug was approved. I think it was over 10 years ago now. And there's been more than 100,000 patients have been treated with these drugs now, and they work. They have their limitations. They don't actually cure. Resistance is universal. But some patients have lived 4 or 5 years on these drugs, and so this was a building block of what we are doing now in myelodysplastic syndrome and in AML here. And I wanted to show you briefly, because this is a clinical trial that we did more recently that was Acute, that was done at the multi-institution, but we led this at Temple. And oh, by the way, this is a CME, so I need to mention that my lab has received support from the company that has developed this drug, guadicitabine. It's called Astex, and that guadicitabine is not FDA approved for the treatment of anything, so that this is still investigational at the present time. And I also didn't mention that I'm a consultant for a company called Daiichi. Daiichi has no epigenetic products, so this is not relevant to my talk today, but for CME purposes, I need to mention that. So guadicitabine is a dinucleotide of decitabine and deoxyguanosine, and this dinucleotide essentially makes it more stable, more resistant to cytidine deaminase, and it's a prodrug for decitabine, and the company wanted to bring it to clinical trials. And so partnering with Stand Up to Cancer a few years ago, we designed a phase 1 study where we wanted to apply the principles that we had learned in the clinic so far, which include the fact that we were not interested in the highest possible dose of the drug. So we did this study, which was quite involved, where the patients were treated with the drug every day for 5 days, or they were treated once a week, and we collected blood before treatment the first day and then every week after that, and we actually measured DNA methylation, and most of this was work in my lab done by Woonbach actually, and we measured DNA methylation almost in real time. That is, we treated 6 patients and then we stopped the study, we measured DNA methylation, then we treated another 6 patients, we stopped the study and we measured DNA methylation, and this was a fascinating result. So at the lowest dose, we saw a little bit of decrease in methylation, but really not much. As we increase the dose, we now see more decreases in methylation, and then methylation recovers because we stop the drug. We give it for 5 days. This is day 30. So when you stop the drug, methylation starts recovering here. Now notice what happens as we increase the dose all the way to 60 milligram per meter square. We get this nice demethylation now of about 25% of the genome, and it goes back towards baseline. When we increase the dose more, from 60 to 90 to 125, we do not get more demethylation. We hit a plateau in demethylation, and that's the phenomenon that I told you about, that as we go up on the dose, we start losing the DNA demethylation effect because the cells do not divide anymore. Now, we didn't continue going up. If we had gone up significantly, I actually think that we would have seen even less and less and less demethylation, but we had designed the trial to stop when we observed this plateau in demethylation. And even though this drug was actually tolerated at 125 milligram per meter square, we decided to take it to further clinical trials at 60 milligram per meter square, which is half the dose we actually give to people, and we could have gone up even more, but we just decided not to based on all of the prior data here. We also showed that the weekly was not enough. You really had to give it for several days here. And so, you know, I'm not going to go over the clinical data. This is still ongoing. The phase 2 studies show a lot of responses in patients with AML and MDS, and there's now been about 3 AML trials, and we're still waiting for complete results of these. In this one trial, treatment-naive AML, we reported at ASH just a month ago that in the entire phase 3 study, there was no difference in survival between the old drugs and the new drug. But if patients stay on drug, that is, those patients who received 4 cycles or more, the new drug was better. So again, it goes back to what I told you. These drugs work slowly. So in patients who are able to take them, this is actually a better drug. Unfortunately, overall, there was no better survival, so at least that trial will not lead to FDA approval, but we're waiting for the other trials in this particular case here. This is just an example of how we're thinking about epigenetics and taking it to the clinic. I want to mention that there's now these 2 and 5, 7 drugs that are FDA approved for the treatment of various cancers that are epigenetic drugs. 2 DNA methylation inhibitors, 5 histone deacetylase inhibitors. There are over 30 drugs in clinical trials. Actually, the IDH inhibitors, these 2 are now also FDA approved for the treatment of AML with IDH mutations. IDH, if you recall, is part of the Krebs cycle, and mutations in IDH lead to accumulation of an inhibitor of a DNA demethylase and very large defects in DNA methylation. And these mutant IDHs have been targeted with drugs which are now FDA approved. This EZH2 inhibitor, the EPZ6438, is actually quite active in patients with Hodgkin's lymphoma who have mutations in EZH2, and I think is quite likely eventually to get FDA approved for the treatment of leukemia. So we will soon have more than 10 epigenetic drugs that are FDA approved for the treatment of cancer. So the field has really moved very quickly, or, you know, it seems quickly now. It was actually very slowly when you're in the middle of it. But we went from basically no FDA-approved drugs that was epigenetic 10 years ago to over 10 drugs now. So this, I guess, is progress here. So this is where we are. I just want to a quick shout out for the Coriell Institute. As I mentioned, we're a block down. We're next door to the hospital. We have a number of different things at Coriell. It's one of the world's most used and most important biobanks. Started the biobanks in the 1970s here. We're very active in genomics and epigenomics. We're developing programs in personalized medicine. There have been programs in personalized medicine based on genomes. We're now developing these further based on epigenomes and with a focus on aging further. And I mentioned to you our interest in drug discovery. We'd love to talk to people about our efforts, collaborations, and so on. We're very easy to find. And so if anyone has any interest, please just knock on my office door or send me an email and we can talk about collaborations here. So I'll stop here. Thank you for your attention, and Jaroslav and I will take questions. Yes, sorry. Can we get the lights up, because it's a little dark, so I can't see you very well. But yes.
I have 2 questions. One is, I accept that cancer is an epigenomic disease.
Yes.
I went to medical school. I went to tell about it. I know about different categories of disease. Are there any other categories of disease which smart people like who are involved in this sort of research are growing to understand are really epigenetic diseases, but we never knew it?
Sure. Developmental diseases. There's actually intense interest right now in diseases that, like autism, for example, where we still don't understand why, there's so much autism right now. Genetics do not explain the majority of cases of autism. There's probably a likelihood that a number of developmental disorders are due to defective reprogramming during embryogenesis and are actually linked to epigenetics in one way or the other. I think that a number of the manifestations of chronic inflammation-linked complications are probably epigenetic. A great example that is likely in part epigenetic is diabetes. I mean, diabetes is one of these things. Type 2 diabetes is one of these things that you don't have it when you're young, you have it when you're old. So what is happening in that process is essentially probably an epigenetic link that makes your cells more resistant to insulin secretion and glucose here. It's hard to study, however. We don't really have good cellular models of diabetes to study how it's epigenetics. I'm sorry?
How about neurological disease?
Well, again, one of the fascinating things is that when you and I went to medical school, everything was genetics, right? And genetics was going to explain everything. And then it didn't explain everything, and then everything was SNPs, right? SNPs were going to explain everything. Now, if you go to these personalized medicine conferences and you ask, okay, you study the entire genome and you study all of the SNPs and you ask, what is the contribution of genetics to any given common disease? It's often no more than 30%. Or, so where is the missing? What is missing? Well, the only thing that could be happening really is epigenetics. Now, it's much harder to study epigenetics. It's tissue specific. So, I would need to have a lot of brain to study brain neurologic disorders, and that's not something that is easily done. But in the genetics field, there's a lot of interest in missing inheritance, and that missing inheritance is probably in part epigenetic. And I think if we had the tools to study it well, we would discover a lot more epigenetic contribution to common diseases.
My other question is anyone using large molecules like monoclonal antibodies or small RNA antisense kinds of strategies to do epigenomic modification in disease states?
Most epigenetic writers, readers, erasers are in the nucleus, and so the antibodies would really mostly be at the cell surface, and so the antibodies won't have the effect that one would want. The small RNAs, I mean, they may. I think there has been some interest in that. The majority of people interested in epigenetics are cancer. And the problem in cancer with the small RNAs is delivery. If you want to fix a genetic disease and you deliver your RNA to 80% of the cells, that's fine. But 80% delivery in cancer isn't going to do anything because the other 20% of cells will kill you. So there hasn't been as much effort, at least in the cancer field, for that. But if we discovered an epigenetic molecule that would, for example, be able to alleviate autism, I'm sure that a small RNA might be the way to go in that case, yeah. Yes, please.
I might be wrong, but when I looked over the D-cytabine,
Yes.
molecular structure is pretty close to cytarabine.
Yes.
So, and cytarabine is a pretty old.
Yes.
So does cytarabine have any epigenetic?
No. In fact, the cytabine was developed in the Czech Republic as an RC or cytarabine analog, and they made a number of them. And they were developed in the clinic as cytarabine analogs, which is why they took them to such high doses, because you can give, grams of cytarabine to people. But the main difference between cytarabine and decitabine is that 5 position. That 5 position is different between RC, and that's what makes decitabine and azacitidine DNA methylation inhibitors and RC essentially a synthesis blocker, but not a DNA methylation inhibitor at all. Yes. But you have a sharp eye there. Yes.
Not so much a question, but a personal interest is
Sure.
the cytogeneticist, and I appreciate the fact that you worked in MDS and AML, and I saw one slide had the MLL gene.
Right.
So, in my mind, when I first heard epigenetics, it was always more like gene regulation.
Well, in the MLL case in particular, MLL itself is an epigenetic molecule, right? So the thought is that some of these translocations in leukemias, for example, are going to lead to permanent gene expression changes which are epigenetic because of disruption of the function of an enzyme. Other translocations do not work that way, and so there's, if you just activate MYC or you have a more classical translocation, then you have a completely different mechanism of action. There is an interest in the field as to the relationship between copy number and DNA methylation. On the X chromosome, there's a mechanism that counts the X's, so that in women, if you have 5 X's, 4 are inactive and 1 is active. And it is at least thought that in some cases that mechanism also applies to gene duplications in cancer. So if you go and you have, 5 copies of a gene, it is likely that, at least in that region, many of these copies are DNA methylated and turned off as a defense mechanism. But then there's one gene there, the oncogene, that would be amplified in that case. So that's a little bit of a special interaction between chromosomes and epigenetics in that case here. Yeah, yes?
About, like, for example, in smooth muscle, when it experiences stretch or injury or pathologic insult,
Yeah.
and we see that the muscle de-differentiates back to more of an embryonic type of myosin or whatever.
Yeah.
Do you think there's any role for the methylation patterns?
Well, any cellular identity switch is an epigenetic switch. I mean, whether it is purely DNA methylation or some other epigenetic mark, we didn't have time to cover histones or other things that could be involved in those things. But yeah, I mean, there's no question that in some of these instances, what is happening is you're reprogramming the epigenome, and that's what is causing these switches here. There's also a lot of interest, not just in there, but in the whole stem cell identity thing. Like in some cases, stem cells, like in the gut, they differentiate, every 3 or 4 they replenish or they renew the tissue every 3 or 4 days. And so that's a rare example of a relatively rapid epigenetic change. And because it's rapid, we don't think it's DNA methylation based. And indeed, we have not found many DNA methylation differences between gut stem cells and gut differentiated cells. This is where there are other epigenetic mechanisms that are much more plastic than DNA methylation. But yeah, what you're describing is an epigenetic phenomenon. Right. That's actually work from our lab. We're really interested in that. But we have shown that in colon cancers can differ in their methylation state. And these differences can be accounted in part by differences in the microbiome. And we've actually generated, we've gotten mice from Christian Jobin that had no microbiome or microbiome. And they have very large differences in DNA methylation in them. So yes, that's why on one of my slides, I actually put down the microbiome as one of the environmental factors that could affect the rate of aging. And there's also an interaction with inflammation in that particular case here. Yeah. Yes?
Most of these agents that you have described here seem to work somewhat associated with cell proliferation, right? You need a certain number of cell cycles.
Yeah.
So that obviously applies well to cancer cells. But
Yes.
How about the other diseases where replication may not be an innate feature of the affected cell? Like you mentioned autism, for instance, or some autoimmune diseases.
Yeah. Well, so we are actually studying this, and a couple people in the audience are studying this here. Aging, right? I think that aging of proliferating cells has a different phenotype than aging of non-proliferating cells. So, the heart ages, right? It's not like the heart doesn't divide, but it still ages. But I think that the aging mechanisms in the heart are different than aging mechanisms in, the gut, where proliferation is happening all the time and things are different. And the same is true for potentially non-dividing neurons, that they do age, but they probably age in a different way than dividing cells. The epigenetic changes we see are much, much larger in dividing cells and in non-dividing cells. In fact, I don't think we've not yet been convinced that we see clear age-related changes in DNA methylation in non-dividing tissues. Having said that, autism and childhood developmental diseases are clearly replication dependent because, you know, the brain is developing through embryogenesis and there's a lot of cell divisions there. So if there's been a faulty, for example, epigenetic program that happened during division of neurons during embryogenesis, that would be fixed once the neuron differentiates and no longer divides, but then it will manifest itself at autism. So it is still a replication-dependent epigenetic problem, just that in that case the replication happened in early embryogenesis, and in fact,
Reversible pharmacologically.
it might be. I mean, the problem with epigenetics is that the replication is absolutely fundamentally required for good reprogramming of a cell. You know, and we haven't discovered that with drugs. We discovered that with embryogenesis, right? I mean, the cells, you know, oocytes, spermatozoa infused, the cell replicates, immediately the epigenome is reprogrammed. Now, there are some people in the field who believe that you can reprogram the epigenome without replication. I actually am very skeptical of that. But there are some people who believe that maybe it is possible. And, you know, DNA repair, for example, when you repair DNA, you have to locally open up chromatin and then repair the DNA and then reestablish chromatin. And maybe in the context of repair, you might be able to have some degree of epigenetic reprogramming without the cells actually dividing. But, you know, that would be very focal rather than general across the genome. But it is a problem if we're thinking of reprogramming, whether we can reprogram non-dividing cells is a real issue. Yes. Okay, 7 o'clock. Great. Thank you all.